then it is my pleasure also to introduce the next speaker, which is Peter Mindek. And Peter Mindek, if you don't know him already, is a co-founder and CTO of Nanographics, a Vienna-based startup developing technology for visualization and storytelling. He received his doctoral degree from TU Vienna in 2015, where he also worked as a postdoc in the area of biological data visualization. His research interests include scientific visualization, storytelling, molecular graphics, and software architecture. In today's talk, he'll be telling us stories about telling stories with procedurally generated images. Please, Peter, go ahead.
Thank you very much. Let me share screen. Let me figure out how it works. All right. So I will be talking about telling stories with procedurally generated images. So what are these procedurally generated images? Basically, they are images created by computer algorithms where we have precise set of instructions which are creating the image. We can do different things with it. We can introduce randomness to the picture, or we can control every aspect of such generated image. So the advantage of creating images this way is that we can create very complex things with a relatively short mathematical description of what's going on in the picture. And another advantage that we can utilize is that we have all these numbers there, and we can change these numbers to get different image of the same class, or we can even continuously change some of these numbers to get a continuous animation. So we can do different things this way. We can create all sorts of abstract shapes, and we can show multi-dimensional mathematical functions. Or we can try to simulate reality. So reality of the world around us, it's very well described in mathematics. So if we want to achieve some photorealistic things like photorealistic lighting or some camera effects, or maybe realistic movement, we can, instead of trying to mimic it with the normal tools, we could try to find a formula which describes this phenomenon, implement this formula, and then get a realistic image of it. Now, the question would be, why would we try to depict reality? I mean, we can take a photograph, right? Or so what's the purpose of doing is beyond it's fun. So one good example is in depicting biology. So here's a watercolor painting of the E. coli bacteria made by David Goodsell. So this is showing a cross section of a bacteria. We can see this DNA in the middle in yellow. In this level of detail, we cannot capture bacteria with current technology. There is no microscope which could show it in such detail, and we can go even further than this. We can show individual molecules in such picture, which is drawn by hand, which are forming this bacteria. And so it's very important when we want to communicate our knowledge about things like viruses or bacteria to have an ability to show visuals how these things actually look like, which we cannot do with our imaging equipment. And you can imagine that this is a very difficult task to create such pictures. Not only you need artistic talent, you also need a lot of time and dedication in order to create such an image. So you can see here David painting one of his paintings, and it takes a lot of time. And the problem is that the knowledge that we have about these things changes a lot. And so often it happens that sometimes you're drawing such a picture, and then after you finish, the knowledge is already updated, and the picture is obsolete, and you have to start over. And so in 2015, approximately, we started this project where we try to make a software which would help with creating such images. And it was called CellView, and it was using this other software called CellPack. And so the idea basically was that, I mean, if you look at such an image, so this is again a watercolor painting by David Goodsell. If you look at this image, you see a lot of repetition. These molecules repeat, and he had to draw them manually. So the software which we would make, or this algorithm would create this placement of these molecules automatically. And the artist would still have all the control over the visual mapping of how it looks like, but all these repetitive tasks would be created by the computer. With CellView, we achieved such results that we could create very realistic or realistically composed images, which still have the visual appeal chosen by the artist. And in this case, we were trying to mimic the style of David Goodsell. The thing with biology is that it is inherently multi-scale. There are important things going on different scales, and when you want to communicate this to somebody who is not biologist, you cannot stay on one single scale. You cannot somebody who doesn't know anything about biology show a molecule and expect them to know what it is, or expect them to be able to imagine what this thing does. And so when we want to tell stories about biology, we have to be able to do these things on multiple scales at the same time. So here's an example. This is electron microscopy image of a virus, of the HIV virus. Then we can see some shapes. We can see the overall shape of it. We can see in the middle this structure called capsid, which holds the genetic information. But we cannot see much more. What we could do with a CellView was, and with a CellPack, was to fill these compartments with molecules. We could grab these molecules from the protein data bank, place them accordingly in space, and then create a representation of a virus where we could zoom from this overall scale to a much smaller nanoscale where you can see individual molecules or even atoms. It's a realistic representation to the degree to which we actually know from which molecules this virus is composed. But it's much more detailed and much more easier to talk about molecules where we're actually showing them than it would be with just this electron micrograph. And so we could create these images of viruses, but what is actually important when you want to talk about things like viruses is to put it in perspective with something that is known. So if you want to talk about the virus to a person who never seen one, you have to start somewhere here. So this is a visualization of a virus where it's 1 pixel in this full HD image and placed next to a human hair, so that people get this context of how large a virus is. Because hair is something that they've seen, they can imagine it's very, very thin, and the virus would be this large in comparison. But still, the virus itself is just 1 pixel, so it doesn't really tell us much. So we can also try to show the virus in its environment where it actually lives. It usually doesn't happen to be next to a hair, but what it usually does, it infects cells. So we can show it amongst other cells. We can show how does it actually look like in this realm of tissues and cells where the viruses are doing their stuff. Here we can see these little dots. These are viruses exiting a cell that was infected before. Now they rupture this membrane and are spit out. So here this scale show us kind of where the virus lives, but again there are still just little dots. And so we can also take a look at the surface of this cell, where we can see how these viruses are attached to it. We can see how numerous there are and how they are everywhere, hidden in these folds. And so this tells us kind of why it's so difficult to get rid of them, because it's so many of them, and because they are everywhere. They are very hidden, and they are entering these cells from all sorts of places. And then again, yet still the virus here, it's very, very small. We don't see too much. How does it actually look like? So we can also show the virus itself. This is in this case a coronavirus. We can show this model, which is detailed enough to show individual molecules. We can cut it in half and see what's inside, see how this genetic information shown in red is organized inside. And so we have all these different scales where we can talk about a virus when you want to illustrate it. What we are trying to build is a system where we can create illustrations where all these scales would be represented procedurally, and where we can create illustrations where all these scales will be kind of shown together. So this is one example of illustration that we made with our system. It's a coronavirus entering the surface, entering a cell, and you can see the whole kind of life cycle of the virus. Because when we talk about viruses, we always, like in media, we see this little virion, this little sphere, and that's a virus. But that's not a virus. That's just one part of its life cycle, right? It's called a virion, where the virus is actually living inside of the cell. That's where it's being reproduced, where it's doing a lot of interesting things. And so in this illustration, we can see how these virions are kind of coming closer to the surface of a cell, and they are attaching to this protein molecule on this receptor on the surface of the cell. And then the cell is kind of sucking it inside. We can see one of these virions being sucked inside of the cell here. Then they are multiplying inside of the cell, and then they are exiting the cell by rupturing its membrane. We can see it here in the background. And so with this force perspective, we can now show these multiple scales in the same image. Or another way how to look at this could be this, right? We can see these little dots in the background. One of these little dots is actually this one large virion on the front to kind of show this force perspective. And so making these images, I mean, it's possible to do it manually, of course, but our goal here is to have a system where everything would be created procedurally. And so there are different aspects of different things that we can create procedurally, and we can help the artist to create such a pictures much faster than doing everything manually. And the first thing would be to generate these structures. So how the things actually look like, how they are organized. And before we use this CellPeg, where algorithm would just randomly distribute molecules or models of molecules according to some rules. But now we wanted to have this real-time system which generates it on the fly automatically. So this brings a lot of advantages. Like, for instance, we can generate much larger models because we can only generate them where the camera is actually looking. So this allows us to create molecular landscapes that are way larger than what would fit in the GPU memory. And so this allows us to create much more complex stories as well in the end. Another thing that we can generate this genome or genetic information, which again, it could be very easily described mathematically on different scales. On the smaller scale, it's a double helix. On a larger scale, depending on the organism, it could be creating these super helix structures, or in bacteria or viruses, usually scrambled up the string, which we can generate procedurally as well on every scale. And it's very important in many cases to show this genetic information. So we have algorithm for that. And it also works in real time. It can generate DNA up to the size of the entire bacteria. So a lot of molecules or a lot of atoms connected together. And then another thing that we have a generator for are these lipid bilayers. They form the membranes of all cells and many viruses, and also all these organelles inside of cells. It's two layers of lipid molecules which are creating this membrane, and we have a generator which can take arbitrary mesh and cover it with non-repeating random pattern of the molecules, forming this realistic looking lipid bilayer. Without, basically only thing that we need to do is to create this mesh, this overall shape, and this we can take from microscopy, or we can do it manually, and then we can cover it with these membranes. And so with all these techniques, we created this illustration. I will now show you somehow stylized video of this whole process of how we made this illustration. So one thing that we did manually in this illustration were the colors. We just chose them somehow. I mean, there are no colors in this scale, right? Because the things don't interact with the light if they're so small in the same way as they interact as the big things interact with light. So we invented colors somehow for this illustration. I mean, it's one picture, so it's not just such a problem. But when you have a longer, like let's say even interactive system where you want to interact with these molecules, you can fly around. It would be great if we would have a system to create these colors also procedurally. And so this is the work of our colleagues. They created the system of creating these colors automatically. I mean, you can assign colors manually, but when you do that, and then when you zoom in, for instance, and your scale changes, the colors stop making sense. On the large scale, they show you individual compartments, maybe like in this case of the virus. But when you zoom in to the small part of this image, all the colors are the same. It doesn't tell you much. So you can also color things, like in this case, you see the atoms, you can color the atoms accordingly to distinguish them. But then again, when you zoom out to a larger scale, you just get a salt and pepper noise, and you cannot even distinguish this virus in the middle anymore. And so our colleagues made a system where the colors are calculated procedurally based on what the camera sees. So based on the scale at which a camera is and what is actually visible on the screen, it takes certain pre-chosen color palette and distributes all the items on the screen into this palette so that the colors are utilized as efficiently and as possible, and also that you can distinguish individual things that you are looking at. So when you're looking at different molecules, they will be colored differently. But when you are zooming out and you are looking at different compartments of this whole model, the individual compartments would be different, will be colored differently. And so in this way, the colors, the only choice that or the choice that the illustrator makes here is what's this basic color palettes. That's what they choose, and then the actual assignment of the colors to the molecules is done by the algorithm. And so we can have these predefined scales at which these changes are happening, which depends on the model, and then we can have a meaningful color palette for every scale that we happen to be in. Another thing that we can generate procedurally is abstraction. It's another thing that is important. We always create these molecular models which are very, very detailed, and you can see all the individual molecules in them. But sometimes when you want to communicate some other things, not that there are many molecules, you want to have a more abstracted version of your illustration. And this is what illustrators are doing when they're creating the stories. They know at which level of abstraction they have to work, and then they create the illustration directly like that. What we wanted to do was a way of generating this procedurally. So we made a system which takes our detailed models and they can automatically turn them into a more abstracted versions of them. So you can, this all happens in real time. So you can have your visualization and you can dynamically switch between different levels of abstraction. You can create a simplified model with models when you want to communicate some other aspects. You can leave some of these representative molecules inside of it. But you can see much better these individual shapes and compartments, and you can talk about them much easier in this level of abstraction. So that's another thing. And then the last thing that I will talk about that we can generate procedurally are the animations. And that's because biology isn't static. Biology isn't only structure. It's also about processes, and that's a very important part of it. And these processes that happen, they can be conveyed very easily with animation. In biology textbooks, you often see processes shown in this way as diagrams of individual steps or states of the system, and then some arrows and some information in between them. So what we did was we created a system where these individual states could be described by functions. And then by interpolating these functions, we can procedurally or automatically generate animations of how these processes would actually look like. So here is an example of a microtubule being constructed. We described the state of the deconstructed microtubule and constructed one, and then just interpolated between them. And then we had these two different ways of how the microtubule can be built or deconstructed, and we can switch between the state of shrinking and growing of this microtubule. So this is a structure in the cells, kind of a scaffolding of a cell. And so creating this animation was very easy with this system, because all we had to do is describe mathematically a microtubule, which is just a spiral of one type of protein. And then we had to describe these open and closed states of it. And then that was it. The system did everything else for us. And of course, we can also take a snapshot of the animation to create a static illustration. in any of these states as well. So the overall idea of doing all of these things that we are doing is to have a system where all the knowledge from biology could be integrated together, and the illustrator would have control over the visual aspects of it. So what colors should be there, how should it look like, but most importantly, they would care about, or they would create the stories. They will be creating this very tedious integration of all this data from all these modalities. They would create the stories, and the system would create the visuals for them according to their artistic vision and according to the storytelling that they want to apply. And these results that we create, they are interactive visualizations, so they can be deployed as an interactive web applications or videos, or they can be printed out as pictures. And so all these algorithms that we are creating, all these things that we are doing, they have one ultimate goal of becoming together this one system where such things would be possible. So I will close with one last video that we made last year, where we were working with biologists who wanted to communicate the structure of a coronavirus. And so we use this system of ours to create a short animation which shows you, based on their data, how this coronavirus is constructed. Yeah, that's it. Thank you for your attention.
I was muted, obviously. Thank you so much, Peter, for a wonderful talk. These are beautiful images, and I'm sure also all the participants here are very impressed because there's many questions also. So I'll begin by asking a few of them. First up, we have Alexander Viet, and he's asking, are there any conventions or standards for color assignment in molecular visualization?
So not really. There is a convention for coloring atoms that comes from the days where you had these plastic balls and you were connecting them. So there's this color scheme, but not really for proteins. So they are pretty arbitrary. That's kind of a problem. But also it gives a lot of artistic freedom to artists. So you see all sorts of colors. They usually mean something in this whole concept of this illustrator who created the illustration. But between the images, you can never say if something is red. On this molecular scale, it means something, doesn't that artists like red color?
Right, thanks for that. The next question is by Sophia Beck. She was wondering the same, and who decided that the spikes of the coronavirus should be red, for example, if you have any knowledge of that?
So in this case, I decided that. So there the reason kind of was, what we wanted to show with this blue and pink kind of use was that the virus is not living thing, it's dead. So that's why we use this blue grayish color for the overall thing. But then we also wanted to highlight the active parts which are actually doing something. So the spikes which are actually attaching to the cell, when they are infecting it, we made them purple, and even more vivid was the RNA inside. It was even more red. And that was because that was actually the describing the whole virus. So that was the most active part, let's say, or most important one. So that's how we chose these colors, just kind of the highlight something against this very muted blue and gray shades of everything else.
Yeah, very nice. Next question by Ace Later. Are the visualizations exportable as polygon meshes? Can they be ported to other animation or modeling programs?
So we have several tools that we built. We have an exporter in mesh. We have exporter as a volume. In these visualizations, we always work with this representation of sort of point clouds. or instances. So each molecule is represented by 1 point in the GPU, and then the GPU has kind of a lookup table of the models of individual molecule types. So the reason for this is that we can create this, or we can render this very, very fast, and we can render many, many atoms in real time. And for us, real time is the most important. I mean, I was showing videos now, but what's our core interest is actually interactive visualization. So all of the algorithms that we use are real time, and for that, it's usually easier to work with these point clouds. But yeah, the answer is yes, we can export it as measure, as a volume.
Okay, cool, thanks. Next question is by Alan Rocha. How is the collaboration with scientists, and how can they validate the storytelling process?
So usually it's a lot of communication. I mean, it's pretty much the same as when they are communicating with illustrators who are not biologists themselves. It's a little back and forth. So usually they send us some images, we kind of look at it, try to understand it. They help us to understand what do we see. Then we create some sort of preliminary visualization. They tell us this is completely wrong, it's not how it looks like, and then we refine it. And this is another reason why we want to have these procedural described images, because when we create something like a membrane or whatever, and it's wrong, we don't have to redo it from scratch, right? We just change this one parameter in the application that tell us, yeah, like for instance with spikes, the first model of this coronavirus, I think I put like 500 spikes on top of it, and it was all just randomly distributed. Then the scientist told me, yeah, it's actually like more like 70 to 100 spikes, right? So this was one number in the source code that I changed, and I had immediately new version. But so it's a lot of this back and forth communication.
Yeah, that makes sense. He has a follow-up question also. He's curious how each step of the animation or storytelling is put together. He says it feels like a combination between video editing and procedural modeling, or is it all part of the rendering engine?
Yeah, so this is exactly that VJ part that I very vaguely mentioned. We have these two libraries. One is called Marion, which creates all these visuals, and all these procedural modeling or procedural generators are implemented there. And then we have a VJ, which is a kind of a library. You can imagine it as an Adobe Premiere or some other software that attaches to your visualization tool, and then you can script everything in your visualization algorithm. So you have a little timeline where everything shows up, and all the kind of parameters that are exposed to the API, where you can change everything from this timeline. So yeah, it's working. It looks like a video editing tool. Basically, it turns your visualization prototyping algorithm or software into a video editing thing. And you can also work with this interactively. So you can say this part is interactive and so on. So yeah, it's a combination of the two, but we have it all in our libraries.
All right, thanks. Next question by Farhan Rashid. Are these algorithms to generate the visualization depending on both theory, models, and microscopy images?
So it depends. I mean, with these viruses, we always start with a microscopy image, but in a sense that you look at a microscopy image, then you create all these shapes, either manually or with machine learning. There are different ways how we can do that. And then we populate these meshes with molecules. But in some, for instance, with this, in the case of microtubules, there we were working purely with numbers, right? We just knew that there are this many turns of this many tubulin molecule in one microtubule, and we just described this mathematically. So we didn't look at the images at all. We just created this based on these numbers, gave it to biologists. They say, yeah, looks all right, and that's it. So it really depends.
Cool. Maybe the last question, because we're actually out of time already, from Laura Garrison. Have you thought about creating storytelling presets based on the audience? For example, a lay audience gets a certain color set, abstraction set, and number types of annotations.
Yeah, this is actually very interesting. I mean, we thought about presets in more low-level way, where all of the parameters that we have, they kind of can be stored as a preset, and then we can work with these presets as kind of as a unit. So we can interpolate between them, and then everything interpolates, you know, not in the same speed, but accordingly, so that it looks visually nicely. So we have done these things, but they are very on low level. So if we would like to go in this higher level, where we could actually say this preset is for that kind of education or audience, so that we didn't get there yet. But it's a very interesting problem, and it will be definitely interesting to explore that area as well.
Thank you very much, Peter. With this, we are already at the end of our session, so I'd like to take this opportunity to thank both of our speakers very much. There's a large virtual applause in the Discord, but I guess you don't see that.
Thank you very
by
much.
the way, there's more questions on the Discord, so if you have a chance to join, I'll put the link in the chat, and maybe you can jump in and answer some more of them if it's okay with you. Thank you
Sure,
so much,
absolutely.
Peter and Johanna. Then next up, we have a 15-minute break, and afterwards we're back to learn more about VTK with Will Schroeder. I'm very much looking forward to this as well. Thank you so much.